Hello, everyone. So today we are going to do a lesson, or today's lesson is going to be as the camera resets. So today's lesson is going to be about what DNA codes for. So we've been learning a lot about the structure of DNA, but DNA codes for proteins. Okay, and the proteins that you have determine your traits. So like, for example, I have blue eyes and many of you have brown eyes. So there's a different protein in my eye, or actually less protein in my eye, that makes my eyes blue and other people's eyes brown. Today we're going to learn how the 4 DNA bases, the A, T, Gs, and Cs, code for a protein. And from the very, very beginning of the year, we learned that proteins are composed of many amino acids bonded together. And I just want to make sure that my microphone is working. Give me 1 second. It is, and my screen is showing. So we're all good there. So if you remember from the beginning of the year, proteins are made in the ribosome, which is located in the cytoplasm outside of the nucleus. DNA, if you remember, is located in the nucleus, and DNA can't leave the nucleus. So it needs a helper, okay? And the helper that's going to help it get the message to make protein to the ribosome is called messenger RNA, and for short, we call it mRNA. And its job is going to be to bring instructions from the DNA in the nucleus to make protein at the ribosome. Okay, so here on our paper, we have a segment of DNA. And again, remember that this DNA is located in the nucleus. Okay, and we need to get the message to the ribosome. So the first thing that's going to happen is that this, as I try to make sure it's on the screen, this DNA is going to be turned into RNA. Okay, and remember we learned the base pairing rules. So when we were doing DNA, A went with T, but you guys just did a video and found that there's no T in RNA. So instead of A going to T, A goes to U, and again, I'm making an mRNA molecule. So G goes to C, T goes to A, U, and I can continue with my base pairing rules. Again, remember there's no T's. We're making mRNA here, so there's no T's here. So I'm writing the complementary base based on the base pairing rules that A goes to U and G pairs with C when we're making mRNA. And sometimes I make a mistake. Hopefully I won't. G, A, C, G. And you're going to be doing a lot of these in the next couple weeks. So again, here we made an mRNA molecule. This is an mRNA. The top was DNA. We know that the top was DNA because it contains T. And then we wrote the complementary RNA strand or mRNA strand on the bottom. Okay. Now the process of turning mRNA or making mRNA from DNA is called transcription. And in transcription, as you guys can't see, I'll move it up. In transcription, DNA is copied into mRNA. And that is the process of transcription. Okay. Now, remember, the goal is to make protein. So in the next part of this process, okay, the mRNA is going to move out of the nucleus and it's going to move to the ribosomes. Ribosomes are parts of the cell that make protein to be turned into protein. And this process has its own name. It also starts with a T, and it is called translation. And now we're going to briefly look at the process of translation. We are oversimplifying translation a little bit. So again, the mRNA that I just made needs to move to the ribosome. So I'm going to represent this box as the ribosome. Okay, and what I'm going to do, this is the ribosome, is I'm going to copy and I can barely see it, but I'm going to copy And again, normally we do this on the same paper, but for today, we're going to simulate it moving to the ribosome from the nucleus. So we're going to copy our mRNA that we just made exactly as we wrote it. And I'm going to be very careful as I do it. So U, C, A, U, G, U, U, C. Hopefully I don't miss any letters. G, G, C, C, C. U, G, A, and I'm going to run out of the box, which is fine. A, U, G, A, 2 A's, C, G. And again, you can check for me. I'll check for myself that I didn't miss any letters. G, U, U, C, G, G. 3 G's, 3 C's. It looks good. All right, so I just copied letter for letter the mRNA, okay, that I made from the DNA on the previous page. Okay, so now we are going to make a protein from it. And if you see at the bottom of this page, we have a codon table. Okay, a codon, okay, is a set of 3 mRNA bases that code for an amino acid. And if we take a look at the code, and I'm going to see if I can zoom in a little bit here, because it's a little small. Maybe that's all right. So there are 20 amino acids, if you remember we learned that, and they're given 3-letter abbreviations. So, for example, all these codons here code for the amino acid pro. These guys here are amino acids. Okay. So now, if you notice, there is a special codon. Actually, let me get a different color. And this special codon is called AUG, and it's the start codon. Now, translation, making a protein can't start until you see a start codon. So if you go back to our mRNA, we try to find where we see AUG. And I see AUG here, and I'm going to put a box around it. And then I'm going to put a box around every 3, because every 3 is a codon. And if you run out of letters at the end, that's fine. You'll see what's going to happen in a second. So now I'm going to, using my table, I'm going to match, and unfortunately, I'm going to have to go back and forth because it's just a little bit too low. I'm going to match the codon to the amino acid. So although AUG is start, it also codes for the amino acid MET. And again, here, so MET, and then UUC. So I try to find UUC. Here's UUC. That codes for the amino acid Phe. And then GGG codes for the amino, here it is down here. GGG codes for gly. And then CCC, which is right here, codes for pro. And then if you see UGA, which is right here, okay, that is a stop codon. Okay, and that tells you to stop reading the mRNA. mRNAs can go on for thousands of base pairs. Okay, so each one of these that I wrote in pink is an amino acid, and they bond together. When they bond together to make a protein. So today, after you've taken these notes, you're going to be transcribing and translating them. But transcribing means turning into mRNA. As I get my new paper over here. And then translating goes to the ribosome, and using these code builds proteins from 3 bases in RNA. So just as an example, this is the last thing that we're going to talk about in this video. Let's say you had a DNA molecule that was 21 bases long. So that's 21 A, T, G's, and C's from the start to the stop codon. Okay? If you divide that by 3, that gives you the number of codons. Okay? So 21 divided by 3 is 7. So in our mRNA, our fake mRNA that I made up that has 21 bases, there are 7 codons. Okay? So that means, you say, oh, well, that means that since there's 7 codons, that means there'll be 7 amino acids. But you have to remember that the stop codon does not code for an amino acid. So that means that our protein would have 6 amino acids minus 1, and the abbreviation for amino acids is AA. I can write it out if you'd like. But because one of the codons is the stop codon, the number of amino acids is 1 less. All right, so now you're going to do some practice where you're going to transcribe and translate some more DNA. And obviously, you'll have my help if you need it.